Yeah, so we're gonna go over quickly everything that you need to know for biology paper one. Um, some things will be triple only, and there'll be text on the screen. You'll see it, so it's not a big deal. But besides that, just make sure everything that you see here, you guys make sure you know it, or you're gonna be cooked. So to begin with, we have our light microscope and electron microscope. The only difference that you need to worry about is that a light microscope does not have a very high resolution. Electron microscope has a high resolution, so you can zoom in a lot more with the electron microscope. But the main thing you need to know about microscopes is the actual practical on how to use them. So here's a microscope. You have a light, which is used to have light, a stage, which will have the thing you're looking at on it, and then your objective lens and the eyepiece lens. You can see there's three different objective lenses over here. So you can change it to different zooms, for example, times 10 all the way to times 40. And the eyepiece lens up here is normally just times 10 by default. So for example, if you wanted a magnification of times 400, you would set your eyepiece lens to times 10 and your objective lens to times 40. So overall, you'd have times 400 magnification. These little knobs here, all they do is lower the stage or make it go higher. That's the only thing. You'll need to know how to actually analyze something using a microscope. So okay, let's be a bit cheeky and we'll do let's do a leaf. Why not? So the way to use a microscope is you need to extract a piece from the thing you're looking at. For example, you can take a piece of this leaf and place it on a slide. You don't want to stain it using a dye, for example, just so we can see it better. And then you want to cover it with another slide. So it needs to be in between two slides. And then all you need to do from there is just put it on the stage and zoom to whatever you need to zoom it to. So again, just cut a piece, place on a slide, stain it so we can see it properly, cover it with another slide, and then put it on the stage. The equation for microscopes is this. So we have an equation triangle with image size on top, the actual size here, add in the magnification. So for example, if I had a image that was 4 millimeters and a magnification of times 20, the way to get the actual size would be 4 divided by 20, which is 0.2 millimeters. I'm not going to go into the plants and animal cell stuff because realistically, you should know that. If you don't know what a nucleus is at this point, then well, you're finished. Don't worry about it. Specialized cells, though, you need to know about the sperm, egg, and epithelial cells. So here we have a lot of mitochondria because, of course, the sperm cell needs energy to swim. You have a tail to swim, and you have a nucleus that has all the DNA in it. The only other thing is the acrosome, which allows the sperm to break through the egg to help penetrate it. For an egg cell, the only thing you really need to know is that it has a lot of nutrients inside of it because the baby needs to grow, right? It's kind of a little bit of common sense you should add to it. These are epithelial cells. So when bacteria and different pathogens come inside your body, they get stuck in the mucus, and the cilia over here can waft it up so you can spit it out. You know when you go like that thing, yeah, it's that. So the cilia will waft up mucus to remove the bacteria. So the last thing about cells is bacteria cells. So you can see here they don't have any nucleus. They have a free flowing DNA. So it's not enclosed in a nucleus. And there's also plasmids, which are just small little pieces of DNA. So both of these are DNA. So the loop over here and the small plasmids as well. They're both DNA. And we have a cell wall and it has a tail as well. Or flagellum, you can say. But that's it for bacteria cells. So again, one last time. Nucleus, no nucleus. We have a free flowing genetic material. And plasmids are just small little rings of DNA. So enzymes are proteins that can act as biological catalysts. This means they can speed up digestion inside of your body. So to begin with, we have this yellow substrate over here. This could be, I don't know, carbohydrate or fat, it doesn't really matter. But you can see how there are two different enzymes, the blue one and the green one. Our yellow substrate can only bind to the blue enzyme because their shapes are complementary, meaning they can fit together. Yeah, so the enzyme needs to have a complementary shape to the substrate originally. And you can see with this green one, it will not fit together, so it won't work. So once the enzyme and the substrate come together, the substrate, this yellow substrate, will break down into its products. And you need to know examples for each thing. For example, you need to know what does carbohydrate break down into, or what do fats break down into, or the proteins break down into. But the main thing is the two shapes are complementary to each other. So the enzyme and the substrate are complementary, so they'll fit together, and the substrate will bind to the enzyme's active site. That's an active site. So once the substrate binds to the enzyme's active site, it will break down into its products. The speed of an enzyme is based on its temperature and its pH. So generally, increase in temperature should increase the speed of your enzyme up until a certain point, known as optimum temperature, which is the point where it's working at the fastest speed. So this is both for pH and for temperature, similar. So once it goes beyond a certain temperature, which is normally 40 degrees, the enzyme denatures, meaning the shape of it changes. And as you can see, if the shape is not complementary to the substrate, then it will not fit together. So the enzyme will no longer be working. So the three types of enzymes you need to know about are proteases, carbohydrases, and lipases. And those are to break down carbohydrates, lipids, and proteins. There are some examples of carbohydrates you need to know, mainly amylase, which is used to break down starch. But generally, you should know the main three. So as you can see in this diagram, carbohydrates will get broken down into simple sugars, proteins break down into amino acids, and lipids break down into fatty acid and glycerol. So when it comes to carbohydrates, starch is also included. So starch as well will break down into simple sugars. And the last thing is just about where they're produced. So carbohydrates, proteases, and lipases are all produced in the pancreas and the small intestine. However, certain proteases, mainly pepsin, which is the one you need to know. I know the handwriting's a bit bad, but pepsin is produced in the stomach. So all of them produced in the small intestine and pancreas, but proteases can also be produced in the stomach. So here are the different food tests that you need to know. So the main thing you need to know is, what reagent they're using. So starch, you need iodine, or for sugars, you need Benedict's. And you also need to know the color change. So I'd say just memorize this. That's the only way you can kind of do it. Just remember each one. So the last part of this topic is calorimetry. So we're trying to find out how much energy a piece of food has. So we begin by measuring the temperature of the water, and then we have to burn the food until it's completely burnt out. And we try to find out how much did the water's temperature change by. Once you know the initial temperature and the final temperature, you can find the temperature change and put into the equation of E equals mc delta T. So if you do the mass times the specific heat capacity times the change temperature, that should give you the energy change, which will tell you how much energy that food had before you burnt it. So diffusion is the movement of particles from an area of high concentration to low concentration. But we're not going to go over that because that should be common sense to you. Osmosis, though, is a similar definition. So high concentration to low concentration, but it's water going through a partially permeable membrane, meaning it's small gaps that only water can go through, but nothing else. The only reason why this is important, because we're going to go over the practical later on about the potatoes, and just, besides that, just if you know the definition, that's fine. So here are the different stages of mitosis. So to begin with, in the interphase, all the DNA is replicated. So we should have double the amount of chromosomes after this. Prophase is when the nucleus breaks down and the DNA condenses. Metaphase is when all the chromosomes will now line up in the center of the circle. So obviously the nucleus is gone now, so all the chromosomes will line up in the middle. Anaphase, they will split apart to opposite sides of the cell. And then telophase is when two new nucleuses are made. Lastly, cytokinesis is when your cell membrane and your cytoplasm will fully split apart. So now we have two identical daughter cells. So this is an example of asexual reproduction because you have a single cell or single organism creating two identical organisms. The main use for mitosis is for growth and repair of your cells. So cell division is pretty good until it becomes uncontrollable, and that's where we have cancer. And you don't want that, or you shouldn't want that at least. I'm not your dad, and you can do what you want. So cells can differentiate to make other kinds. So cells can differentiate to become specialized cells, which are just cells with special functions. For example, a white blood cell or an epithelial cell. Animal cells only specialize at the beginning of their life, so as a baby, for example, whereas plants can do it throughout their entire life. You can find animal stem cells in embryos and in bone marrow. So when it comes to embryos, they can specialize into any kind of cell, but with the adult ones in the bone marrow, you can only turn into, for example, blood cells, but it can't change into any kind of cell. Stem cells can be found in a plant's meristem. You also need to know two examples of stem cells being used. The first one is type 1 diabetes, where stem cells can be used to create pancreas cells, which can release insulin. Also, for paralysis, stem cells can be used to create nerve cells so that a person can move again. The last part about stem cells is this, so the benefits and drawbacks of using stem cells. So obviously the main benefits are they can turn into any kind of cell. So it can cure things like diabetes or paralysis. The risks though, I would say you should know one medical risk, one social risk, and one like ethical one. So for a medical one, you could say stem cells can get rejected by the patient, right? The second one is for, let's say, for social, you could say, there's not that much evidence of it being successful because it is quite a new modern technique. And for the ethical one, you can say stem cells are, they can come from destroyed embryos, which can have some ethical problems. So here's a picture of the brain. The main three parts you need to know are the cerebral cortex, which is the largest part, and that's for your conscious things, such as language and intelligence. The cerebellum, which is the small part on the bottom right, that's for controlling the motor functions. So your movement, your arms and legs, just how everything moves. And lastly, the medulla, which is at the bottom, controls your unconscious activities, like breathing and heart rate. To study the brain, we need to use a CT scan, which shows a 3D image of the brain and you can see if any of the tissue is damaged. There's also a PET scan, which will show you the functions of the brain. For example, is your medulla working properly or your cerebellum working properly? You can check that. So CT scan will only show you the 3D shape of your brain, but a PET scan will show you the functions if they're working properly. So here's a picture of the nervous system. So a receptor cell will receive a stimulus, meaning, let's say your hand, for example, it will detect a change in the environment, like the temperature. So once your receptor has detected the stimulus, it should travel up the sensory neuron. So the impulse will take it up the sensory neuron to your CNS, which is your brain. Your CNS will process the information and then send it down the motor neuron to your effector, which will cause a response. For example, your brain can send a message to your arm muscle for it to contract and move out of the way so you don't get burnt, right? Because you don't want to get burnt, hopefully. So yeah, that's the main pathway. So stimulus gets detected, impulse gets sent through your sensory neuron to your CNS, and your CNS will send the impulse through the motor neuron to the effector so I can respond. Now, if it's a reflex, the only difference would be that instead of going to the CNS, it'll go to relay neuron instead. So, for example, let's say you touch something that's burning, right? You'll detect that it's burning, and the impulse is sent through a sensory neuron to the relay neuron to the motor neuron. So there's no CNS involved. Instead, it'll go straight through the relay neuron to the motor neuron. It's also important to know what a nerve cell looks like. So the long part of the neuron is called axon. So this whole thing's axon. And the main thing you need to know about a nerve cell is that it's surrounded by a myelin sheath. So the myelin sheath is an insulator. So it makes sure that the electrical impulse that's going through the axon is insulated. So that makes it faster. So the myelin sheath is an insulator, which will make the impulse go faster in the axon. That is the main adaptation in a neuron cell that you need to know. Also, dendrites, they branch out a lot because they're trying to connect to other neurons. So if you knew what dendrite is and you know about the axon and the myelin sheath, that's all you need to know about a neuron. So again, axon is this long part. It's insulated by the myelin sheath, which causes the electrical impulse to go faster. The last thing you need to know is how do two neurons connect to each other? So you can see here, these are two different neurons, and we need to see how does the impulse travel from one neuron to the next one. So once the electrical impulse reaches the end of the first neuron, it will trigger the neurotransmitter chemicals to diffuse across the gap. So this gap is known as a synapse. The neurotransmitters will then bind to the receptors in the next neuron, and this will trigger a new electrical impulse to carry down the second neuron. So again, electrical impulse goes to the end of the first one. It triggers the neurotransmitter chemicals to diffuse across. These will bind to the receptors of the next neuron and cause a new electrical impulse to be triggered, and it will travel down the neuron. So next up, we have the eye. So next up we have the eye. So obviously there's a lot going on here, but you don't need to know like 99% of the stuff. The ones you need to know are the cornea, the iris, pupil, the lens, and the retina. So cornea is the outer layer and it bends the light. That's all you need to know. So it's the outer layer of your eye and it should bend some light. The pupil is the hole which allows the light to come inside. Your iris controls how big your pupil will get. So if it's a bright light, your iris will cause the pupil to close. Or if there's a very little light, your iris will cause your pupil to widen and open up so you can take in more light. At the back here, we have the retina. So the retina, we need the light to hit the retina for us to actually see an image. So the retina is at the back of the eye, and we need it to see the image. The lens will focus the light onto the retina. So light will just come in through the pupil like this, and this blue section here, this lens, will focus the light so to make sure it hits the retina. If an object is like really close, then your lens will get thicker to try bend it even more. For example, if you had an object that was right here, the lens will get thicker and bend the light harder. If an object is really far away, your lens will get thinner and refract it less. So if it's really close, it should get thicker and refract more. But if the object is really far away, your lens will just be thinner and refract it less. So some people are nearsighted or long-sighted. So if someone's nearsighted, meaning they can't see things that are far away, it's because their lens is too thick, so it refracts things too hard. If someone's long-sighted, it means they can't see things that are close because their lens are too thin. It doesn't refract it enough. Again, if someone's nearsighted, it means that their lens is too thick, so it refracts it too much. So you want to use a concave lens to counter that. So if someone's short-sighted, you need to use a concave lens. If someone's long-sighted, you need to use a convex lens. And lastly, we have cones and rods, which are in your retina. So cones will detect different colors, and rods will detect light. And that's the final part of the eye. The two types of reproduction are sexual and asexual reproduction. In asexual reproduction, only one parent is required, and a genetically identical offspring is made. But in sexual reproduction, two parents are required, and genetically different offspring are made. So one advantage of asexual reproduction is, it only requires one parent, so it's very easy and quick to do. But a disadvantage would be if it's an exact clone, the same problems that will affect the parent will affect the child. For example, they'll both be susceptible to the same diseases. With sexual reproduction, it is more difficult and it will be slower because it requires two parents. However, all the offspring will be genetically different, and because there's a lot of variation, they are more likely to survive. Sperm cells and egg cells are known as gametes, and to make gametes, you need to do meiosis. So meiosis is the process of making gametes, and they should only have half the chromosomes of a normal cell. So a sperm cell will only have 23 chromosomes, whereas a normal cell should have 46. So this is DNA. Before we continue, one important thing you need to know is protein is made up of amino acids. So if you had a chain of amino acids, that makes a protein. A gene is a piece of DNA which codes for an amino acid. That means if I had let's say this section here, so A T C G, so this whole section here, this codes for a specific amino acid. A bunch of amino acids together makes a protein. If someone had blonde hair, for example, they would have a set of these genes here, which will code for some amino acids, which makes the blonde hair protein. So genes code for specific amino acids. That is what a gene is. And this is the structure of your DNA. You have your sugar phosphate backbone, which is this section over here, and it's over here as well. So this is the sugar phosphate backbone, and you have the bases in the middle. These bases are hydrogen bonded together, and you need to know that. So it's a hydrogen bond between them. A always matches with T, and C will always match with G. So these are the bases in your DNA. So that's the most important thing. DNA is made of genes which code for amino acids that can make protein. All of the DNA of one organism is called a genome. For example, all the DNA in every single human, that's a human genome. Or if you got all the DNA from every cat, that would be the cat genome. You also need to know how to extract DNA from a fruit. So these are the steps over here. So you have to first mash up the fruit and then add detergent and salt. You have to add these so the cell membrane can break down so the DNA can actually be released. You then want to filter your mixture and then add ethanol. So ethanol will make the DNA actually visible. So before you add ethanol, you can't see any DNA. But after you add the ethanol, the DNA will be visible. So this diagram here is showing protein synthesis. So obviously the diagram, there's a lot going on, but it's not as complicated as it looks. So to begin with, your DNA will unwind. This makes the genes like available for us to copy. So all the DNA should be in your nucleus. So for us to take the actual DNA out, we need to send in an mRNA into the nucleus to copy all of the DNA, and then they can exit the nucleus. So the DNA itself will never leave. The mRNA will come in, copy the DNA, and then leave. The mRNA will now go all the way to the ribosome. Once it binds to the ribosome, every three pairs of base, so every three bases will produce one amino acid. For example, let's say we had A, T, and C, that will produce a single amino acid. And when I say produce, tRNA will bring in the specific amino acid based on those bases. So if I had an ATC binded to the ribosome, tRNA will bring in the amino acid that codes specifically for that. Once we have enough amino acids, they will become a chain and fold to make a protein. If there's a mutation in your genes, the protein that is made will be different. This is because the mRNA is copying your DNA. If your DNA has changed, your mRNA will copy the wrong information, which means the protein that it makes will be completely different. Different versions of a gene are known as alleles. For example, you can have a blonde hair allele and a brown hair allele. There are dominant alleles and recessive ones. So a dominant one means you only need to inherit one of them from your parents to have that characteristic. Whereas a recessive one, you need to have two to inherit the characteristic. This combination is called a genotype. For example, if you guys ever see something like A and a small a, that is your genotype. A phenotype is the actual characteristic that is being expressed. For example, blonde hair. So AA is the genotype, blonde hair is the phenotype. So this can be represented using a Punnett square. So we have two parents, one with capital T alleles and one with small t alleles. So a capital letter means it's dominant and a lowercase letter means it's recessive. And again, you need to have two recessive alleles to experience the actual characteristic. So in this case, for you to be a short plant, you need two recessive alleles. But to be a tall plant, you only need one of these capital T's. So dominant only requires one of them. So you can draw a Punnett square to show which genes the actual offspring will inherit. And I think most of you should be able to at least fill out the Punnett square. And you notice that there's capital T small t, capital T small t, capital T small t, and a capital T small t. So these are all going to have the tall characteristic shown because tall is a dominant characteristic, so you only need one of them. If we had a small t, that would give the short characteristic. But in this case, you have 100% chance of having a tall offspring in this case. You need to be able to write as percentages as well. So for example, if one of them did have small t, you would say 25% would be short and then 75% would be tall. This offspring has four heterozygous alleles. This means that its alleles are different. For example, it's capital T and small t. Homozygous means they have the same allele. So a capital T or small t. Charles Darwin's theory of evolution is all about natural selection. So the first important thing you need to say is there is a variation in the population because of mutations. In this case, the snail shells have different colors. So some of these mutations would lead to the animal lasting or living for a longer time, which means they're more likely to reproduce, which means their genetics get passed on. So this is natural selection. Selective breeding is when you pick two animals with desirable characteristics and you breed them together. But obviously, that's not enough. After they make offspring, you have to breed the offspring together in order to make more offspring with the characteristics that you want. So it's always breeding two of them together and then breeding the offspring together after as well. You can't just do the first one. So genetic engineering is a process of changing the genes of an organism so they can have desirable characteristics. To begin with, you need to cut out the gene that you want using a restriction enzyme. You use the same restriction enzyme to cut out the plasmid in the bacteria cell. You use ligase to stick this gene into the plasmid, and then you reinsert the plasmid back into the original bacteria. So now this bacteria will have the gene that you wanted it to have, for example, the insulin gene. Communicable diseases are diseases that can be spread, and they're caused by pathogens. So the four pathogens you need to know are bacteria, virus, protists, and fungus. And here are a few examples of diseases that each of them cause. I would say you should know at least one for each. So you should know an example of a virus, like HIV. You should know an example of a bacteria, like tuberculosis. So each one, you should know one example. And non-communicable diseases cannot be spread. And here are some factors that might cause them. So smoking, obesity, and alcohol. So when a virus infects your cells, there are two different pathways it can go through. In the lytic pathway, the virus will infect your cell and use the cell to create more of the virus components. It will then explode and release the virus particles around to infect more cells. In the lysogenic pathway, again, it begins with the virus injecting its DNA into the cell, but this time, instead of exploding, the cell will replicate and create more virus cells. Afterwards, it'll go back to the lytic pathway. So the lysogenic one will eventually lead to the lytic one as well. So to defend against different pathogens, you have stomach acid, which can kill bacteria, or even your mucus can catch pathogens and you can release it from your body. However, the main thing you need to know is how do your white blood cells protect your body from pathogens. So there's two types of white blood cells. There's phagocytes and lymphocytes. So here in this diagram is a phagocyte, and what it does is it will engulf the pathogen and digest it. And here are lymphocytes. So lymphocytes release antibodies. These antibodies will bind to the antigen of the pathogen and destroy them. So each antibody is specific to antigens, similar to enzymes, where the lock and key, where they have to fit together. So a lymphocyte makes antibodies specific to each pathogen. So this is how vaccination also works. So in vaccination, a pathogen that's dead or weak is introduced to your body. Your lymphocytes will then produce antibodies specific for that. So and the memory cells will remember how to make the antibodies. So that means the next time the pathogen enters your body, the lymphocytes already know which antibodies to use to destroy them. We use antibiotics to kill bacteria. One important example that you need to know is penicillin. You also need to know how were these drugs even developed to begin with. So these are the three stages of developing new drugs. We have the preclinical trial, where we test the drug on cells and to see any side effects. We then test it on healthy human volunteers, again to check to see if there's any side effects. And for the final stage, we do a double blind test, which means we give some of the patients a placebo, and even the doctors don't know which ones were given a placebo, just to make sure that the results that we get are perfectly accurate and aren't biased in any way. So plant diseases normally cause spots to grow on the plant. This means there'll be less chlorophyll, which means less photosynthesis, which means stunted growth. The last two diseases you need to know are HIV and coronary heart disease. So HIV kills your white blood cells, which leads to AIDS, which means your immune system is fully compromised. As for coronary heart disease, this is when the arteries in your heart are clogged. The two ways to treat it is by using statins or stent. So statin is a drug which will lower your cholesterol, which will make the build-up in your arteries decrease. A stent is something you insert into your arteries. It's a balloon which will blow up and create more space so that blood can flow again. Monoclonal antibodies are a way to mass produce antibodies. You begin by injecting a mouse with a pathogen. The mouse's lymphocytes will then produce antibodies specific to the antigen. You take the lymphocytes from the mouse and fuse it with a tumor cell. This tumor will uncontrollably divide and create a lot more of these lymphocytes, which means we have an endless supply of the antibodies. And these aren't just for viruses or bacteria. This could be used to detect hormones. The antigen could be a hormone, for example, a pregnant woman's hormone. So it's not only just for diseases.